surrounded to this particular veins. So because uh, because because this is the very highly imitated to the particular resource. So then again then again then again started to the particular waves and particular regardings. So then again the formation of new breeds. This is the one of the next one irrigation cultivation though. Formation of new breeds, how to the new breeds growing of this particular step. This first step in the silkworm breeding process in primary evolution of intel breeding materials like that breeds lines hybrid is easily. Usually the following evolution is made. So some some characters are some particulars are depend by this particular phenomena. So mainly how many types and the sources. So like that this is following the evolution. What is the evolution? Evolution means the separate of some particular species and a particular variations. The phenotypic evolution of the main qualitative and quantitative breeding characters. This is the first one following the methods of um, phenotypic evolution. So, phenotype means is the structure and formation of the one of the characters of in insect. This is the two types: phenotype and genotype. Genotype is the genetical characters decided by the genetical side. Phenotype is the structural of insect characters. The first one, phenotypic evolution, is the main qualitative and quantitative breeding characters. So main important first evolution is the phenotypic. The next one, phenotypic variations of the values of the quantitative characters. So variations, how many variations of phenotypic? So for example, Bombas mori species, the genotype is 3,000 above the global wise. The global wise 3,000 above species of genotyping is there. So because the first evolution is the phenotype variations of the values in the quantity of characters. The next one, hybridability of main quantity of characters. Hybridability means the hibernation. Hibernation is the incubation, which is the eggs are incubated. The eggs are good quality and good productions are the, it is the only hibernation of the specific eggs. So it is called as hibernations. It is quantitative characters. The next one, correlation between the main quantitative breeding characters. Correlations, what is the correlation? The correlation is the statistical report. The correlation, negative correlation and positive correlation, it is depend by the specific the specific zone and specific specific insect families. So this is the only mm, correlation is the negative correlation as positive correlation. Which is the animals are positive correlations and negative correlation. The depend to the one was the specific variations, because the correlation is identify the standardization species like that Bombax mori and Muga, Tessar, Isar, so like that. So because the mainly the following of evolution, this is the one of the character. The next one, regression between the main quantitative breeding characters. Main quantity of breeding characters. The breeding characters is the main regression between the main quantitative breeding characters. The next one, for breeding new silkworm race from the different methods are used. How many different types of used for breeding silkworms race? The mainly segregation from foreign F1 hybrids. The first one, segregation. Segregation means is the crossbreeding. Segregation is the one of the crossbreeding characterization. Crossbreeding means is the by the method is Bulgaria were selected most of the older silkworm breeds like virtual one or two. So this is the foreign F1 hybridization used to the Bulgaria country. The generally the methods are followed. Mainly five types of methods are followed. Segregation methods. The initial population. The taken the first one initial population. It is the generation of first generation F1. F1 means is the first generation of one. It is called as the one of the hybrid. The initial population. The next one selection of larvae having markings and elongate shaped cocoons in F2. F2 means generation 2, second generation families. The next one, maintaining the selected mass. The maintaining the selected mass. Mass is the silkworms of life period of fourth stages. The next one, the next one, batch rearing selection for larval makings, cocoon shape, texture, main quantitative characters, and for the 11 to 16 generations. So 11 to 16 generations. So why? Because the 11 to 16 generation used to. This is the only because the species like that more than important more than important up to the specific characters like that use the specific characters of initially like that the particular phenomena. The next scenario is the separation from the foreign F1 hybrid. How to separate? The methods follow the separation methods are initial population, South Korean hybrid, and selection of cocoons having the elongated with construction shape and maintain the between them breeding of which batch breeding, rearing of selection of cocoon shape and main quantity of characters. So then next one, auto breeding are 9 generations selection for cocoon shape and main quantitative characters and a new breed of the isolation of this particular. The using population of the Japan race. Japan race, how to the using of Japan race? The same follow the this scenario. The first one segregation and the segregation separations of this uh, particular species. The next one very important of authorization of new breeds, which is the species of authorized because the authorized species of segment to used the irrigation and getting the producer main silkworm fiber threads. So because this is the main important of this particular particular position. So then next one, uh, the authorization of a new brood race development by CSR and TI Mysore. The Mysore is the one of the breeding produced laboratory because this is under of CSR, Council of Scientific Research and Technology of India. So it belongs to location at that Mysore. The followed the particular process. The first one niche traditional of poly hybrid pure Mysore. Then tradition of polyvalent and bivalent and multivalent. So this is the use of the particular race development. The polyvalent breeds and hybrids of the pest pest development by to Mysore Princess, Osa Mysore, Tamil Nadu White. Then this is the particular species names. Then next one, early Powerland Pure Race and Kolar Gold, Kogel Javan, Mysore Princess, Osa Mysore, Tamil Nadu White, Osa Mysore. So this is the particular names of authorization of new breeds names. Then next one, next one, uh, this is the fresh cocoons held by one box of eggs, 2000 eggs in 1 kg, in calculated by the following formula. Fish fresh cocoons held by laying 2000 into number of larval counted. The filament weight weight or grams in the one found after the cocoon railing. The filament is derived content weight and washed, white and washed. So this is complete depend by this particular this next unit of unit third, heterosis and hybrid vigors. Heterosis and hybrid vigor. So mainly objectives is the heterosis hybrid vigor. The mainly objectives heterosis hybrid vigor are not or outbreeding enhancement is the improved in increased functions of many biological quality in hybrid offsprings. So mainly nowadays of hybrid offsprings are used by the particular phenomena because because the result maximum genetic contributions is a part of parents. The part of parents of contributions are to specific identification and specific zones of some particular reasons. Then again, those effects can be due to Mendelian or non-Mendelian inheritance. So mainly the sericulture functions, the sericultures or cultures are depend by directly or indirectly genetics methods, genetic methods. So genetic methods, then process of the genetic specialization of the first generation and second generation. So complete this is the particular variations. So so the sericulture depend by the mainly directly or indirectly, indirectly, genetic genetic performance is the main because the Mendelian saying the only one structure. The segregation law is the main important because the hybridization depend by the segregation formula. So then next one, the hybrid vigors important to activate our surveillance of hybrid offsprings. So the mainly hybrid species are surveillance depend by the regulating of temperatures, regulating of tidal waves, and regulating of humidity. So this is the main important of growth of hybridization species like that. For example, Bombax mori and Muga, Eri, Tesser. So this is the main scenario. Depend by the hybrid species of surveillance, depend by the directly indirectly regulating of temperature and regulating of humidity, then vital gases also. So then according to Lee, the phenomena is a heterosis is the reverse of inbreeding and is a depend as a restrictions of vigors. But according to law, one of the American scientists, name is Lee, 1993, is choosing the phenomena heterosis. So what is the phenomena heterosis? Heterosis main is the multiple characters of inbreeding to the specific species, like that hybrid species. It is the name of the heterosis. Heterosis. It is called as heterosis. The point explaining a performance to for the parents. The first generation, F1 generation, it is the first generation. The hybrid generation is F2 generation, the very multiply developmented and multiply organs are developmented because this is the growing and production and best quality and cocoons are having the F2 generation. This is the F2 generation and expanding the performance of the parents' genes. The next one, the less related The less rate of the strains in the more heterotic the reactions is this is can be demonstrated of crosses. Demonstration demonstration mean is the correlations. What is the correlation? The correlation belong to negative correlation and positive correlation. The specific species, this is a correlation saying the good quality species and good production of the silkworm species. This is called the group of families, leaving to the group of families surrounding the particular areas. Then next one, the however, when to inbreed few lines are crossed, the dominant favorable alleles suppress the appearance of effect of the reserves and superior in results. This explanation does not exclude the possibility of complementary gene action. The complementary gene action, the complementary gene action to affected by heterosis depend as the extra vigors are superior to shown by the F1 pregnancy over either it is the parents of the main parental values is given by the formula. The formula used and choose the particularly uh, formula is F1 mid parental value by mid parental value into 100 or F1 better parental value divided by better parental value into 100. So this is the heterosis formula because identified and choose the particular species genes are used to the used and solve these problems are used by this formula. Then this is the heterosis formula. The next see observe the hybrid vigors. The hybrid vigors are how many characters are decide this is the six characters are decide the hybrid vigors. The first one, duration of breeding. So the silkworm's lifespan is a little bit very low, a very little less of time, only 45 days. So the characters are decided by the first one, duration of feeding. How many days of feeding of first instar caterpillar and second instar caterpillar and third instar caterpillars. So the first character, duration of feeding. The next one, larval mortality. So each species find out the main value of mortality and natality. Mortality means of leaving of birth and leaving of caterpillars is very important of larval mortality. So next one, the characters is next one, double cocoon rates. Some species like the hybrid species of double cocoon rates are like the one of the best characters identify the hybrid vigors. The next one, silk filament size. Each filament size length and size at the same time thickness also. So main characters of the this is the economical process of and this is the very identify the character of cocoon filament size. The next one characters of hybrid vigors, cocoon shell weight. The shell weight is important. The shell is formation by CaCO3 calcium carbonate. So this is the weight of shell weight. Best quality silkworm shell weight is a best production of silk fibers, silk threads. So this is one of the consider of this particular characters. The next one, a number of eggs. The next one character is number of eggs are main important. The how many members of eggs are eggs of there the particular reason of this important to this character. The hybrid vigor refers to the positive heterosis. The hybrid vigor choose the only positive positive heterosis. It is the extra vigor or improvement of performance show the first generation pregnancy over either its parents or mid parent value. So because the hybrid vigor also the influence to the heterosis because the first generation genes are convert to the hybrid to the second generation F2 generation. F2 generation is the parental alleles. Alleles is the growth of it is short time and complete the lifetime is complete only short time. Then next one, this phenotypic expressions is also dependent upon the genetic resourcement of organisms. So each and families are each and animals. So each and animals are DNA structure and genetic is saying the character one generation to another generations. So parental characters same to the F2 generation also, because the genes, the study of genes, one generation to another generation, same character for performance to this particular uh, zones of a organism. The extra vigor is shown by the outcross between two inbreed lines of silkworm could be due to the extra buffering capacity of resulting organism. So some hybridization species are accepted or either not, because interesting of genes calculation like that to phenotypic and genotypic. The phenotypic is the structurally divided to the all species names, and genotype is the internal. Animals are accepting and some species are accepting or not. Why? Because the mainly reason is phenotypic and genotypic difference between to the phenotypic and genotypic. So during the different stages of silkworm development, my characters have been found to have a relationship with the qualitative and quantitative aspects of silk yard. So mainly the sericultures are depend by the quality silk yards are the irrigation silk irrigations are depend by the best location environmental distress, like that environmental ecosystem choose the conditionally temperature conditions and humidity conditions and gases conditions are very important. So the indirectly directly the affected to the specific zone of temperature zones and isolation zones and tropologic zones. This graph is mainly divided by the best quality of yield and the particular ecosystem places. Then, however, the characters which relay in terms of manufacturers of heterosis also following the specific characters like that. First, the mortality rate is the lower than the other parents. So mortality means is the birth of instars. The it count the main count to the parents alleles. The next one, the double cockles rate is the higher than rate of the parents. This is the one of the F2 generation. The F2 generation means is the how many higher than the rate of parents. This is a calculation of one of the character. The next one, the cocoon weight is the higher than that the MPVE. MPV means of mid parental values. MPV mean mid parental values. The mid parental values of cocoon weight and higher. The next one, cocoon's shell layers are however than that mid parental values. So then next one, the length of silk fiber is longer than that the mid parental value, and the cocoon fiber weight have higher than the MPV mid parental values. So this is the conditions to study the hybrid vigors. Hybrid vigor, not a hybrid vigor. Hybrid vigor is one of the entertain of heterosis cultivations. What is the heterosis? Heterosis culture is the development of a species. It is one of the name as the hybridization. Heterosis is a different crosses systems. The crosses systems is mainly first one generation F1, F2, F3 generations. So this is the main important of hybrid vigors and heterosis, hybrid vigors and breeding. Enhancement is the improvement of increased functions of any biological qualities in the hybrid offsprings. The next one, heteros is the depend as the extra vigor or superiority shown by the first generation pregnancy over either its parents of the mild parental values. The next one, the hybrid vigors are the phenomena of heterosis is fully exploited in silkworm and maize breeding. So already maize breeding is one of the nutrition food activities and gives the nutrition so that like that the particular small inches. In the case all other animal breeding, pure line selection methods in made use of. The next one, the hybrid vigor refers to positive heterosis is the it is the extra vigor of important in performance shown the first one generation or either it's a parents of mid parent value. So the first one generation, this is mainly uh, heterosis hybrid vigors are mainly depend by the correlations because positive correlation and negative correlation. The correlation is saying the mortality values and generation to the change the second generation growth of second generation like that parental genes to allele genes to the F2 generations produce the common. This is the MPV. MPV means mid parental values. The calculation of segregation mid parental values. So this is the heterosis and hybridization and how many methods of hybridization and how to the carrying the heterosis values, how to the finding the formula, use the formula, then mortality of cocoons and hybridizations of laying of eggs. Then uh, next one, uh, next one, parental race. The parental race is the parental race and distributions. The parental races and distributions based on origin of place, based on volatinisms, based on multinisms, the main important. So learns the importance of parental race. The parental race, the understand about the distribution of race. The next one, now about the volatinisms and multi-volatinisms. The volatinisms or multi-volatinism, how to the growth and produce the parental race. The very important. The history of a silk is old as the history of mankind, mankind, and so is the history of a silkworm. So each and everyone, he knows that this economy, agro industry economy, agro industry cultivation, he knows that Asian countries like that, China, Japan, and India also. So it is the world as the history of mankind, and so is the history of silkworm. According to archaeological and biological graphics evidences, it is a probable that the sericulture was practiced in the China about 2500 BC, and Bombax mori is a kind of wild silkworm were raised in the North China and guessed the repeated selections of purification by humans for a long time has become the silkworm today. So this is the each and everything that the first one the cultivation find out the China the year of 2500 BC. The name of species of Bombax mori. Uh, so then again next ASEAN countries are developed before Japan, European states and India. The different climates and areas produce several regions specific silkworm race with different characteristics due to geographical isolation for long time. On the basis of native regions, the silkworms for four types. So mainly the silkworms are mainly four types. They are Japanese, Chinese, European, and tropical race. European race are the native of European and Central Asia. Tropical of India race are native and India is the Southeast Asian. The unusual Univalent, bivalent, and the multivalent race are found in Japan, China, Europe, and India. But some of those variants of like multivalent are popular of warmer varieties of reached in India. So multivalent breed is very important because it is the yearly growing of at the year of two times of three times of growing this multivalent species and multivalent cocoons are. However, the parental race confine the particular geographic region in the involved a certain combine the value of the newly variations. So because of the particular geographical area, why? Because choose the particular areas. The particular areas of depend on the ecological climates or ecological conditions are the particular geographical areas. Like that, summer climate is different, winter climate is different, and rain climate is different because the summer climate is a highly temperature produce a highly temperature. The silkworm temperature growing of best temperature 25 to 28 degrees Celsius only for growing of best temperature of silkworm species. So then next one, parental race. There are different races in silkworms. There is classification based on native regions, volcanism, multinism, cocoon color and shape, larval marking, the number of hatching year of rearing period, body markings, cocoon color and egg colors. So this is the mainly important of the particular European Valentine, univalentine, China univalentine, Korean univalentine, China bivalent, Japan bivalent, tropical polyvalent, Japanese univalent. So structurally, color and uh, and marking of larval. So those are classification by the parental race. So based on places of origins. This is the one of the geographical geographical study of this particular areas. The based on place of origins, where it is the find out the first one geographical places or first one places of which is the origin is developed the particular health or particular location. There are four types of race. There are follows the first one Japanese race, Chinese race, European race, and tropical race are Indian race. So these are four types of based on place of region places: Europe, Japan, China, and India. So this is the places of particularly uh, region to the birth of origins. So next one, the based on uh, volcanisms. Uh, Volcanism is the ability of a silkworm to produce one to several generation in a year. Based on volcanism silkworms are broadly classified into three types. So silkworms are barely classified to three types: univalentine, bivalentine, multi or polyvalentine. So here are the details of different volcanisms in silkworms. What is the meaning of and what is the structural difference to between univalent and bivalent and multivalent? So univalent silkworms are producing or one generation in a year are called an univalent. Univalent silkworms are generation only 1 year called as univalent. So bivalent they they produce two generations in a year. Univalent is the generation in a 1 year. The bivalent produce the generation of 2 years. The multivalent is the produce more than 5 to 6 generation in the year and suitable for the tropical region. Mainly suitable of a tropical region. It is belongs to the tropical regions called to the Indian race. Indian race belongs to the multivalent and multivalent race. So the multivalent race are they produce more than the particular region of the particular uh, uh, particular areas. So So multivalent produce more than 5 to 6 generations in the year. So multivan is one of the most important economic characters along with the Waltinisms. Along with the Waltinisms also, including this particular characterisms. There are at least five different type of molding in the silkworm, namely di, tri, tetra, penta, and exomeltars. So at least five different types of silkworms. So the most common type in the domestic silkworm varieties and is wild moss or tolerances. Whereas the almost all parents are commercial strains are tolerant tetravalent. Tetra malting characters has been considered as the malting characters is also affected partly by the sex-linked maturing genes. So the dominance relations are mainly M3 plus M greater than the M5. So this is the those parental race are distribution the different geographical parts. There are Japanese, China, European, and India race. Race are classified on the basis of native organism and volatinism, cocoon color, and larva markings. The race can be differentiated from another two basis of morphological, biological, and commercial characters. So most of species are different by this particular morphological, biological, and commercial characters. Japan's race like non-hibering eggs. Larvae grow slowly, black in color slowly. Eaters have inferior cocoons. So Maltanism is a racially characterized based on which is the silkworms also classified into three groups. They are tri malters, tetra malters, and penta malters. Tri, tetra, penta. So this is the silkworm groups of cultures. The next one, breeding stations. So breeding stations are mainly silkworm seed organization, basic and multiplication centers, and cocoon rares and seed crops, cocoon markers, and seed unit of breeding stations. Then tomorrow I will explain the this class. Okay, if we any doubt, make the chat box and uh, okay. Thank you, one and all. Thank you. Thank you.